In our recent study, we explored the oncogenicity of mutant p53 proteins by distinguishing between silent and oncogenic mutations. The following video is devoted to the topic, Method for Predicting the Oncogenicity of Mutant Proteins, using the example of p53 protein. Our study devoted to this topic was recently published. Let's consider the most interesting results devoted to the separation of silent mutations from oncogenic mutations of the p53 protein. You can read this work by the links under the video. TP53 mutations data were extracted from 4 non-overlapping databases, and variants found at least one time in each dataset were included in the cancer shared datasets. For the negative set, protein variants that were never found or found only once in human cancer have been selected. No cancer p53 variants. In this analysis, only missense variants have been included as they are the most common alterations detected for TP53 and the most difficult to classify. Variants were labeled for the positive set and negative set using well. Let's move on to a description of the data obtained. Fig. 1 shows the results of the calculated data for the positive set of p53 protein mutations, which are presented in blue and red. The results for the negative data set are depicted in black circles. Fig. 1 processed calculated data for the negative and positive data sets for the calculated value of Kd. Blue and red indicate the results for the positive set of mutations, and black indicate the results for the negative data set. Note that negative values of the Kd value correspond to a Kd value less than 1. So we are talking here about the values of the dissociation constant corresponding to values less than 1 mole in the positive range of values. We obtained such calculated data as a measure of change in entropy, a measure of change in stability, a dissociation constant for monomeric p53 protein containing different types of mutations, and plotted the resulting graphs for a clear visual representation. The graphs show the difference in calculated characteristics for positive and negative sets of p53 protein mutations. For neutral mutations, we see minimal fluctuations of values around the mean, while for oncogenic mutations, we see a large spread of values around the mean. The graphs show the results for the following calculated values: dissociation energy and a measure of entropy change. The following graph contains information on the change in enthalpy for different types of p53 mutations. You can subscribe to our channel and watch more great simple overview videos from the world of molecular science.